Okay, so inside the cytoplasm, we have ribosomes all throughout it. We have ribosomes on the endoplasmic reticulum, on the rough one, but we have some that are just floating around. And this is why. So they're floating around to connect to and hold a messenger RNA. So then we can transcribe it. So it took its information from the DNA, and now it's giving it through our transport RNA. So messenger RNA copies down the message. The ribosome will hang on to it, hold it in place, and a transcription RNA will then transcribe it. Take a copy of the copy so it can float off somewhere else. It can all connect together, creating a new molecule for that cell. Again, giving that cell its information of what it needs to do. Come on. We really need to watch this video. Because this stuff is a little hairy. Can be a little hard to understand. Which there will just be, a few questions on it. Not very many. Come on. Finally.
One of the main functions of DNA is to provide information for building proteins. Proteins are the body's main structural molecules. They also contribute to almost every cellular function. The manufacture of proteins occurs in two main phases, transcription and translation. The transcription phase begins when the nucleus receives a chemical message to make a new protein. At that point, the segment of DNA with the relevant gene unwinds. An RNA enzyme then assembles RNA nucleotides that would be complementary to the exposed bases.
So messenger
the nucleotides
RNA
attach to the exposed DNA and then bind to each other to form a strand of messenger RNA, mRNA. The length of messenger RNA actually consists of a series of 3 bases called triplets. Each triplet, called a codon, is the code for one amino acid. Once formed, the messenger RNA separates from the DNA molecule and moves through a nuclear pore and into the cytoplasm, where it begins the process of translation.
Out of the nucleus into the
In the
cytoplasm,
the messenger RNA attaches to a ribosome. The ribosome moves along the strand of messenger RNA reading the codons. Waiting in the cytoplasm are transfer RNA, tRNA, molecules. Each transfer RNA consists of 3 bases, a triplet called an anticodon, that will perfectly complement a specific codon on the messenger RNA. Attached to the transfer RNA is the amino acid for that site, according to the genetic blueprint. The transfer RNA finds the 3 bases that are complementary to its own and deposits the amino acid. The ribosome then uses enzymes to attach the lengthening chain of amino acids together with peptide bonds. When each triplet has been filled with the correct amino acid and the peptide bonds have been formed, the protein is complete. The largest known human protein, Titin, consists of 33,000 amino acids.
so all of that work inside the nucleus, the DNA unzipping, the RNA going in there and transcribing just that section, the messenger RNA, leaving there, attaching to a ribosome, and then giving its information transcribed here by an amino acid, ending up with a protein. It took all that to get a protein. So remember that a chain of amino acids will give rise to a protein. So yes, ribosomes make proteins, but it's a long process. Okay, this is the very last part of our lecture. Thank goodness. The cell cycle We're going to touch on the cell cycle. We're not going to go into it in depth because we'll do this again when we talk about the reproductive system. So you do want to know what's going on in each of these phases of mitosis. So this is where we're not doing meiosis where we have sex cells coming together and creating a brand new cell like you are a bunch of cells put together and you're a brand new organism. No, we are making a copy of cells. So you have this cell and we made a copy. So it'd be like your skin. You want your skin to be the same as it was when you were 2. You don't want it to be changing, which it does as we get old, and making copies over copies over copies start to fade after a while. So that's what happens with your skin too. Huh?
Is that what happened with little
Michael?
Michael?
Yeah. No. But he had a skin, disorder. No. It's a disorder. There's a problem with the DNA in the cells. There's a mutation. There's a problem. Okay, so we're starting out with our prophase. So we've got our spindle fibers. They're starting to appear. You still have your nucleus, but now the chromatins are starting to come together with the cytomere, which is a little dot in between there. So they're starting to look like chromosomes. The chromatins that were just all throughout here are now starting to look like chromosomes. They've got a defined little cytomere, and they're looking like a chromosome. So we get into metaphase, where our spindle fibers are going to the polar opposites, attaching to those cytomeres, lining all those chromosomes up so we can rip them in two. Chromosomes, remember that was our DNA information there. In anaphase, they get ripped apart, pulling them to opposite poles, and then we end up with our daughter cells. We split off, you have half the cytoplasm over there, half the cytoplasm over here, and we have the DNA. I should have loaded this video while I was doing it because it's going to take about 10 minutes to load. So you do want to know each one of those phases for this exam, but I just want you to know what's going on in the phase. Again, we'll get into in-depth in it when we get into the reproductive system. We'll look at mitosis and meiosis. So mitosis, we should have the exact same copy. If I start with a liver cell, I want to end with a liver cell.
A key focus of a cell's life cycle is mitosis, when the cell splits into 2 identical daughter cells. Mitosis consists of the following 4 phases: prophase, metaphase, anaphase, and telophase. In prophase, chromatin begins to coil and condense to form chromosomes. Each chromosome consists of 2 strands, called chromatids. Each strand contains a single molecule of DNA. The 2 chromatids join together in the middle at a spot called the centromere. While the nuclear envelope dissolves, centrioles move to opposite poles of the cell, and spindle fibers form in the cytoplasm. This is the final stage of prophase. During metaphase, some of the spindle fibers attach to one side of the chromosomes, and the chromosomes line up along the center of the cell. The next stage of mitosis is anaphase. The centromeres divide, forming 2 chromosomes instead of a pair of attached chromatids. The spindle fibers pull the newly formed chromosomes to opposite poles of the cell. Each pole will have an identical set of genes. Telophase is a reversal of prophase events. A new nuclear envelope develops around each set of daughter chromosomes. The spindle fibers disappear, and the cytoplasm divides to produce 2 identical daughter cells. The growth of organs and tissues in a developing child, the repair of damaged tissue after an injury, and the replacement of cells that die through the course of everyday living all involve mitosis. The only cells that don't divide through mitosis are sex cells.
So you'll have some areas in the body that will do a lot of mitosis. So if you think about your skin and you're constantly shedding that skin, it will go through mitosis very quickly. And the same thing inside your mouth is still your skin, and we'll learn that with the next lecture when we do integumentary system. So think about when you get a sore in your mouth or you bite your tongue. It heals pretty quickly, a lot quicker than even out here on the skin. Whereas other things that we don't rough up and tear up pretty regularly does not go through mitosis as quickly. No, we don't. Let's go change. So that's it for Chapter 3, and we'll do Chapter 4 tomorrow.